and stuff. So just a quick background check. So we're talking about cell-to-cell signaling, and cell-to-cell signaling becomes relevant in the context of evolution when cells become aggregated to form organisms, what we call multicellular organisms. So I mean that's the reason why most of the cases that we're going to talk or discuss of cell-to-cell signaling are actually placed in the context of development of tissues and organs. That's really where you should be talking about cell-to-cell signaling and how it evolved and what kind of mechanisms became available for living organisms to talk between cells. Now, today we're going to approach a very specific case, but I think it's a very good case study because in the period of 4 or 5 years, I mean, people actually came from just describing a pattern of an organ with a certain number of tissues to really understand how different cells get positioned in certain places by mechanisms of cell-to-cell signaling. And this is the root pattern information and the maintenance. Actually, there's a mistake. There's typo here. Sorry about that. And the maintenance of a stem cell niche, and this is a problem which is transversal to any kind of multicellular higher organisms. How do organisms maintain stem cell niches? Because they need to maintain stem cell niches to make new cells or to differentiate new cells when they need to. Most of the research that I'm going to talk to you about were actually supervised or coordinated by this gentleman here, Ben Sherris, a Dutch scientist, then working at the University of Utrecht. Now he's in Wageningen, but also in the Netherlands. And pretty much the concept we're gonna try to approach is like there are niches of stem cells in all multicellular organisms. In plants, I mean, pretty much you see them in a few organs, like the apical meristem, the root meristem, but in plants, rather than animals, I mean, any cell at any point can redifferentiate and dedifferentiate. So in theory, any cell can be potentially the precursor of a stem cell niche. So how come plants are not just big balls of cells which are like basically going around in the fields and become so differentiated and so organized? So that implies that somehow the plants has ways of position the stem cell niches in very specific locations and restrict them there so that you don't have stem cells everywhere and you get differentiation where you need to have differentiation. Does that make sense to everybody? Okay. Yep.
I just have like a general question about all of this. So like, these are like plants, right? So is the thing that we're trying to relate to humans, is it about how like stem cell differentiation?
No, we're just talking about cell-to-cell signaling. I'm just using this as a proxy of a specific biological example in which cell-to-cell signaling is of essence so that the right cells get positioned at the right position and with the right differentiation. Okay? That makes sense? I mean, what I'm trying to say is that like what you should keep in mind is the concept of cell-to-cell signaling is essentially for the right organs to develop in the right way in the right position. Okay? And that's done through cell-to-cell signaling. In the case of auxins, that's very easy to illustrate because auxin is normal, so it diffuses between cells in ways that we discussed on Tuesday. So you can easily grasp it, and that's why I'm using it as an example. Okay. I'm not pretending to turn you all in greens or make any kind of plant proselytic discourse. I'm just using this because I think it's a very good example of cell-to-cell signaling. Okay. And in many respects, actually, more advanced than the examples I could, more advanced in our understanding of the molecular mechanisms than any example I could give you on animals. But we will get to animals at a point, okay? If you guys allow me, you don't make me a lot of questions. And by this I'm provoking you because what I want you is to make me a lot of questions so that we don't advance that much. Whatever we advance, you guys understand, okay? Okay, anyway, let's skip the embryo and let's go to the root. Why the roots? Well, roots are a great organ to study cell-to-cell signaling because they're so very well structured and there's a limited number of tissues, so you can actually reduce to this 1, 2, 3, 4, 5, 6 different tissues in terms of differentiation. So you have the vascular bundle, you have these orange cells here that we call the pericycle, then you have these blue ones which we call the endodermis, these yellow ones that we call, the cortex cells, and then we get into the epidermis that goes all the way up, and then we have these two specialized tissues which only happens on the tip of the root cells, which is the lateral root cap and the columella. And these two tissues only form on the tip of the root because the root has to penetrate through the soil to the soil. So this is like a helmet that the root has in order to be able to penetrate through the soil and not get disturbed. Now the cool thing is that actually we know that the meristem, in other words, the stem cell niche, is around that area. And come to think about it, there's a group of cells here that I didn't mention, which are these brown cells that we call the central cells, that people really didn't understand what they were doing there. So it came as a bit of a surprise that when people look very precisely to these cells, like in the 90s, they found out that these cells never divide. These central cells, the ones which are labeled here as brown. And people call this the quiescent center. Why? Because these cells never divide. And that is like, so you have two cells in the middle of what is supposed to be the stem cell niche that never divide. Why? How come? I mean, what is the role? What are they doing there? Why don't they divide? Is this some sort of a positional marker, like a kind of two cells that are stuck there to say the other ones around them like do something else and kind of define that this is the point in which all cells are generated? Because we know that it's the case. I mean, all cells, all the division that happens in the root happens here, around this area. So what you see in the upper parts of the root is just differentiation and division and elongation. There's no more definition of cells. So all the differentiation takes place here, at this point, around these central cells that never divide. Okay? So people start wondering, why are there? And many times, I mean, what we find as a very useful way of, at least in biology, because our experimental degrees of freedoms are not like the ones that people have in chemistry or physics, in which all the elements can be known. So what we can do as a first approach is be very brutal with the organisms and start by either mutating genes or killing cells. Okay? So let's kill these cells and see what happens, or let's kill cells around it and see what happens. How do they differentiate? What kind of cells they become? So how do you kill cells in an organ? Okay, anyone has an idea how you can kill cells in an organ? Okay, so you can kill them genetically. Like, for instance, we spoke about promoters, and I'm going to speak a lot of promoters also a lot during this class, because I want you all to realize that when we kind of been able to assign that the gene definition, as we spoke here on the last class, basically if you take it like as a promoter and as a region which then becomes transcribed as a protein, this is a fantastic leap forward, because if you have the right promoter, say like if you have a promoter which is specific for genes that only express on the epidermis, you can express any protein on the epidermis and specifically on the epidermis, just by doing a transgenic organism with a construct that has a promoters from the epidermis and whatever protein. I don't know if any of you ever came across these rabbits that glow, that have GFP on the epidermis, but that rabbit was not produced by a scientist, because you don't know. It was produced by an artist. It was an artist performance. I mean, like they make a glowing rabbit. But of course, the science behind it is solid. So this guy picked up an epidermis-specific promoter, hooked it to GFP, and then generated a transgenic rabbit that glows. And that rabbit was in the stand on an exhibition. It was not used for any kind of scientific purpose, other than just the artist feeling happy for creating something which he thought aesthetically was beautiful. And let's not discuss the ethics of doing art this way, please. Because I have thoughts about it. Anyway, if you know a promoter for a specific cell or for a specific tissue, you can attach a protein that may be a toxin. Say, call her a toxin. Say, if you instead of putting GFP in that rabbit, you put call her a toxin. I mean, you would have basically rabbit without epidermis, which is not a very nice picture and probably wouldn't happen because it would die before getting to any significant age. But this is the concept I'm trying to tell you. So like if you would have like a promoter which is specific for these quiescent cell centers, you could put a protein which is toxic like RNAs or a toxin and kill specifically those cells. So you can do that genetically. But most people do it like the brutal way. You shoot where you want to kill, okay? And how do you shoot cells? You shoot with microscopes. Everybody heard about confocal microscopes? So confocal microscopes can focus a lot of light in one single plane. The thing that usually people don't tell you is that these lasers are huge beasts. For normal imaging, we're taking advantage about 1% of the power of the laser. So if you crank up the laser like to 20% or 30%, you'll literally fry whatever you're imaging, okay? So you can go down there and just basically focus at a specific cell, crank up the laser, kill the cell, and see what happens, okay? So these guys did it like in the 80s, and pretty much they came out with 2 papers in Nature that I'm going to describe to you. Always confused, this is the one. And one of them, they went into these cells here, the ones from this kind here, the pericycle and vascular bundles. I mean, the color coded in this scheme is the same. So we're talking about these cells here. And basically they asked the question, because notice this green cell, they have an intermediate stage in which they are not really differentiated, but when they divide, they differentiate into this blue one and the yellow one. So somewhere in this pathway from the greens to these ones, I mean, they have to decide, what am I going to be? Am I going to be a cortex or an endodermis cell? And you can ask the question, what determines that? And there's 2 general models. One is lineage. So a daughter cell basically becomes what the mother cell was already. Or there's position. Whatever happens in an organ, there are certain positions in which cells receive signals and information from neighboring cells. And basically, what defines cell fate is position. Now, early in the 20th century, people thought lineage is the important thing. And in certain cases, that is the case. But for most cases of development, position is the relevant thing. Lineage is the mother cell that tells the daughter cell what to be. And I'm going to tell you the experiment. So this guy here killed, let's use, now that I managed to do it, let's use the cursor. So this cell here, he killed it with a confocal. So the way in which this cell is going to be substituted is the one which is next to it is going to divide to occupy that space. Now, if it would be a lineage, you would expect that cell to be orange, right? But what happened in real life is that cell is going to be green. In other words, it's not the lineage, it's the position that defines what the cell fate is going to be, which forces you to think this can only be by cell-to-cell signal. In other words, I mean, like if I kill an epidermis cell, I mean, the cell which is going to substitute it, it's going to be an epidermis cell because it's receiving information from around it, not just because of the lineage, if you take this literally. Does that make sense?
Yeah.
Okay. So, I mean, through a number of these experiments, they came up to the conclusion that, one, it's the position, not the lineage, that determines cell fate. So it's going to be cell-to-cell signaling. And by other experiments that I'm not putting here, not to complicate, they came up to the conclusion that the information was coming from up. Because if they kill all the cells up of that one, then that was lost. While if they kill all them around, that was not lost. So some cell-to-cell signaling stimuli was coming from the upper parts of the root to determine what was the position and how cells should differentiate there. Okay? Everybody following me? Yep.
We're talking about the differentiation of the cells
Yeah, we're talking about this green cell. I gotta get discipline to do this with the mouse. This green cell was killed, so the cell which is going to divide to occupy that position is the orange one. But when the orange one divides to give a daughter cell, instead of doing an orange cell, it does a green cell. Okay, and this is very clear because in this case specifically, the endodermis are highly differentiated cells, so you can immediately see that that was not a cell that gave birth. Okay. Okay, so just to give you an example of what I just said, when you use the laser of a confocal to ablate things, so this is an embryo of Drosophila, and what people did was they target this cell here on the embryo, and they crank up the laser and see the result. I mean, basically they punch a hole into this cell. So this cell was completely dead. So you can go to an embryo, and you can specifically kill these cells and ask questions about all the cells around it, or what's going to be by that space, or what's going to be the fate of that cell, etc. And you got hundreds of examples like this on the list. The next thing they did, of course, and that was even more interesting, was let's ablate the quiescent cell center cells, because those are the ones that seem to be static there. Why are they there? Why don't they divide? And more interesting, if you kill them, what's going to happen? Are their position going to be taken by one of the cells around? Because if their position is to be taken, one of the cells around it has to divide and take that position and stop dividing. And I'm not going to show you all the experiments. I mean, for those of you who are interested on that, I can send you the paper and we can discuss. But there were 3 models that they were testing with these ablations, killing cells one by one. Does the quiescent cells go up, go down, divide from the left, divide from the right? And basically, pretty much, what they could come to the conclusion is that the quiescent cells inhibit the differentiation of the cells around it, and because the cells around it are indifferentiated, which by definition is what kind of cells? Stem cells. So the cells around the quiescent center are kept as stem cells because they are inhibited by something which is coming from the quiescent center cells that stops them to differentiate. And because they're not differentiate, they continue to divide. So it's the cells next to it that actually start differentiating. So you have this absolutely beautiful scheme. You have a few cells. It's not just 2. This is a section. Actually, I think it's all 8 altogether. The quiescent cells, they never divide. They, by some cell-to-cell signaling, tell the cells that surround them, don't differentiate, divide. And it's the ones that they divide, the daughter cells that they divide and then starts differentiating to give rise to the 8 types of cell types that you have there. So the patterning of the root actually is as it is because this is all coming from the center and cells are differentiating and dividing in all directions, in different ways. Okay? So 2 questions. What keeps the cells of the quiescent centers quiescent, and what is the signal that they use to signal the cells around them to, first, do not differentiate, second, divide, okay? And if we go up to the root from differentiation, division, then comes also elongation so that the roots can grow, okay? So let's get back here. So now we know that the quiescent cell centers, what they do, their role is to inhibit the other cells from differentiate, and what they do is basically these cells around here. So now you can define the stem cell niche as basically this group of cells. Quiescent cell center and the cells that surround it, because they all can at any point divide to give cells that differentiate. Okay? Why is it positioned in that location? Why is kept in this static kind of scheme? And of course, by then, this was before DR5. Everybody is still in tune. What is DR5? We discussed it Tuesday. What is DR5? Come on, guys. It's not a transcription. It's a promoter that is sensitive to auxin binding proteins. That we can use. If we have a promoter which is sensitive to auxins, what can we do with it? You can what? True, and?
We can use it to determine which cells are from.
Right. We can put a reporter, let's use the right terms. You guys have to learn how to use the right words when you get to the exam and for your future life. We have to use a reporter. So if we hook up a reporter to a promoter, that reporter will report whatever activates the promoter. The promoter is a promoter which is specific for auxin binding factors. Then the reporter will report the activity of auxins. Okay? Yep.
So in this picture, it's the cells immediately around it are supposed to be undifferentiated, right?
Yes.
But these look like they're differentiated.
Yeah, well, that's basically for mechanical constraints. But you can, on the basis of molecular, signs or molecular reporters, you can say that they are indifferentiated and at any point, and that's what they prove with the ablation, they can return to it. So if you come here and you ablate a quiescent cell, It's not one from below that differentiates in the nucleus cell, it's one from the top. Okay. So, yeah.
So the ones below the quiescent cells are not stem cells?
For a reason that I will explain you next, because that actually relates to the question, what positions the quiescent cells center there? Okay.
So it's from now.
It is a position, it's not a lineage. Okay? I know this may be confusing, but guys, I mean, this is how organisms develop. And I can give you examples on C. elegans, on Drosophila or stuff, or humans. I mean, well, we don't know about humans, but we can extrapolate from these other animals that probably is the same. Okay, so when people develop ER5 and start doing this kind of localization of auxins, it became more or less obvious that you have a maximum of auxins on the quiescent center. Okay? So if you remember one of the first slides I put about auxins, what happens to growth when you put a lot of auxins, like more than the optimal point? It goes down. Instead of promoting division and growth, it inhibits growth and division. So people start thinking, okay, so we have a maximum peak, which, you know, it's really on top of the auxins of the quiescent cells. Could it be that, like, really what's happening is there is kind of a pattern of the localization of the pins or whatever that brings a lot of auxin to the quiescent cell, and that's what marks the position. Okay, that's what defines the fate of these cells. It has a lot of auxins there, and these cells cannot divide. Therefore, and because in the surrounding cells there's a little bit less of auxins, then the cells are also keeping from differentiating. If auxin is being read like a lot of auxin, no division, a little bit less of auxin, no differentiation but division, and a little bit less of auxin, division and differentiation, and a little bit of less of auxin, growth, just enlargement. Okay? Because that's what it would look just by simply looking at these images. But looking is one thing, proving it's a different thing. And what people went about to do is actually do a number of genetic experiments. This is Cell paper. I don't know if you guys ever heard about this journal called Cell, but you know, it's like, you know, the journal that if you publish a paper in Cell during your PhD, you have a job, basically. Because that means that you really went to the top of the people that can actually stick their nose into this journal. And that doesn't mean that you will not get a job if you don't publish in Cell. Don't take me literally on that, but let's say that it's at this level of nature and science, but a little bit more respectable for people that actually do cell biology. And what they did was a number of experiments in which, by using various mutations of various auxin transporters, for instance, pin 1 here, and this is cool because now you see that pin 1 kind of moves the quiescent center a little bit up. Okay, so the absence of these auxin transporters changed the position of the quiescent cell center. And more interesting, I mean, these cells that you see here, like pinkish or reddish, these are cells from the columella. This is a very specific cell marker, which is the accumulation of starch that can be easily staged. And what they do is when they make it in a pin mutant and other mutants, they clearly change the boundary of the location of these cells. So, in other words, what I'm trying to say is just by affecting the way in which auxins move from cell to cell, they could change the position of the quiescent center, and they could change the position of the cells around it. In other words, they changed the position in a way in which the stem cell niche is actually regulating the growth of the root. So in other words, they could prove that whatever puts the stem cell niche and defines the growth of a root, it's dependent on auxins. Okay? Does that make sense to everybody? Okay, so next experiments, I mean, I'm going to show you all the experiments, but the conclusion was the following. So during embryogenesis, I mean, because the pins get located in a certain way that they create foci or points or spots in which there's a lot of auxin. These spots in which there's a lot of auxin will give rise to a quiescent cell center, and to a quiescent cells, and these quiescent cells basically organize a stem cell niche around them. Either during embryogenesis, either when they do ablation with a laser, they go there, kill the cells, and the ones next to it, which is the one on the top, as I just told you, divides to give a new quiescent cell, and in that case the columella cells gain a new layer. Or in the case of flooding the root with auxins from the outside, then you got diffusion of auxins, you got auxins all over the place, and you got the definition of new quiescent cells and the definition of root stem cell niches. In other words, these roots are very strange. I've got to put the picture because they start growing in every direction instead of just growing down. Okay? So effectively, just by changing the position of the auxins, you can change the cell fate of the cell, and you can change the growth pattern of the organ. Okay? Now, we're coming to, yep.
What if you ablate or change the auxin concentration after embryogenesis?
Yeah, these are these experiments basically here, and the ones that I'm going to show you in the next couple of papers, because that's what they do.
Right.
Okay, they do that because the DR5 shows up.
Mm-hmm.
But meanwhile, I mean, everybody started doing genetics with all the pins.
Mm-hmm.
I told you there's 8. Forget about the 8 because it's only on pollen. But the other 7 people had localized them on the root, and they knew they were not in the same place. They were very specifically organized in position.
Mm-hmm.
And this is precisely the next slide. So same team, like 1 year or 2 years after, and this is exactly what they did. So they went to see where is pin 1? It's here. What happens to pin 1 when you express it in the background of a mutant of pin 3, 4, and 7? It happens like this. The pattern changes. Okay? Pin 2, where is pin 2? Pin 2 is here. It's localized. I show you these pictures. It's localized on the bottom of these cells and on the top of these cells. What if you express pin 2 in the background of a pin 3, 4, and 7 mutant? It totally changes. It tells you that pins are organized to form a certain pattern. Okay? And what is this pattern? I mean, it's schematically shown in this diagram. You have, I mean, so the color of these arrows reflect the position of the pins and what we can deduce of the flow of auxins from the position of the pins. So if all is correct, and we're talking about not only the localization on the wild type, but also how they change when you express it in the mutant. And what you have is what we call reverse fountain pattern. Auxins go down by the action of these 3 pins here, pin 1, pin 2 mostly. They get here, and pin 4 does the job of concentrating the auxins on the quiescent center cells. And then from the quiescent center cells, and this is the thing that makes the quiescent center cells, is that they have pins all over the place, so they kind of redistribute the auxins in every direction. So what you have here, it's a problem that any plumber could tell you how it works. So you got all the pipes bringing the water to a kind of a deadlock. It's not a deadlock. And then the water kind of escapes in every direction. Why does it accumulate there? Now you can tell me. Just like if you were a plumber, could you predict that there's going to be a lot of pressure of water in these quiescent cells? Why? Yep. I mean, you can simply predict that if you admit that the auxin that comes there, it's more than the auxin that they can kind of leak in every direction. Okay? Very simple. So let's put this in a better picture. So this is what you have to keep in mind from like these 10 years of research. You have this cell-to-cell signaling mechanism based on auxins. Auxins are transported by pins. Pins are differentially localized on the cells. Some are on the top, some are on the bottom, some are on the sides, and we can deduce the direction of flux of auxins by saying where is the pin. If the pin is on the basal, then the pin goes down. If the pin is on the top, then auxins go up. Okay? If pin is everywhere, auxins go everywhere. And you have this thing. The quiescent cell center cells have a very high concentration of auxins. Then the cells a little bit below of them, the columella cells, have less. And this answers your question. What's your name, sorry? This answers your question. I mean, it's the one that on the top that divides it, because even so it has much less auxins than the one below. So it's more prone to divide than these ones, because it has a little bit less auxin. And then you have around the quiescent cell, you know, kind of a gradient of auxins, and this kind of dissipates as it goes up. So you have a reverse fountain of auxins that kind of go up. And if you go a little bit up on the root, you can actually see that this is a closed loop. I mean, when it comes here, it goes down again. Okay? So we look at this and solve the problem as if we were plumbers. Yeah.
How do you know how much concentration of oxygen?
We don't. This is just a reporter. We cannot quantify what is the concentration of. And this is a very good question because it allows me to get to the next part of the story. We can look at this and say intuitively, we solved the problem because we're plumbers. This is just a problem of plumbing. Okay? You have pipes. Depending on the diameter of the pipes, there's going to be a lot of water coming there. If you don't have enough reflux, I mean, the water is going to get stuck there and kind of basically being redistributed wherever you have pipes. Okay? But this is thinking intuitively like plumbers. Can we actually test this hypothesis? And can we actually answer your question? Does this make any sense in terms of real concentrations of oxygens? And this is where you get a point in which cell biology and genetics does not give you an answer. Okay? And you know, just this is the image that I want you to keep in mind. Okay? This is the one. That's why I put it a little bit more blown up. And the answer came from a completely different field or perspective. And since this was already introduced by Marowitz in these videos, I think it's important to tell you guys that this is really a big deal. If you're going to do research, or even if you go to medical careers or any sort of careers, I mean, this is a major trend and it's only going to get bigger, so you should be prepared to that. We're talking about the fact that we know from physics and chemistry for many years that the way that these guys actually solve problems ever since the 19th century is actually by going into mathematics. Trying to define things mathematically, try to extract the laws of nature, and then from the laws of nature, try to understand whatever phenomena they see on nature. Now, biology is rather complex. I mean, if you read the text that I put here, this is a paper that came out in the 80s, before all this started. I mean, people were already making these questions, why physicists-like models and why biologists should, and actually the answer for that is actually on the first paragraph. There's no other system with a level of complexity like the simplest cell. I mean, the simplest cells you can get have like 300 active genes, which means that they have at least 300 proteins interacting to make a cell, which means that you have 300 variables on the system. There's no system in physics and chemistry that has 300 variables, not even 30. Okay? So the degree of complexity that we're dealing, it's much higher than normally physics and chemistry. Yes, computers cannot do it, and we did not have enough information to do it. Turns out that in the beginning of the 2000s, the situation changed when we started to have the genomes. So now we know all the genes. And now, I mean, 10 years pass or 15 years pass, we know all the transcripts, and we're starting to know all the proteins. And guess what? The computers, I mean, like, went on like as a power law. So the computers, this computer here, I mean, like the first mainframe I had in my university would fill this room and do like a tenth of what this computer is doing, or any tablet that you guys are using. So the capacity for the computers to actually do stuff went way above. So what happened? I mean, there was a movement that was kind of the guru of this movement, this gentleman here, Hiroiki Kitano, a Renaissance man. I mean, he was an architect, a designer, a biologist, a computational biologist, really a very inspiring person. And he kind of coined this term, systems biology. So what is systems? Now, I mean, the reasoning was, we've been extremely successful in explaining a lot of biology by breaking it in parts. This is the time that we put all the parts together and start looking at the systems as a whole. That's why it's called systems biology. And this issue of science, as you can see, was published in 2002, was kind of like, the beginning of the movement. I mean, the movement was already there, but let's put like, that's when this became a buzzword. So if you want to get a grant approved by the NIH or the NSF these days, if you can crank up the word systems biology in it, there's a very good chance that you're going to have more money than if you don't. Okay? Well, of course, you should have a good reason for doing that, not just putting the name there. Okay, so how does it work, or how is it conceptualized? So we have all these omics. In this case, I mean, in 2002, it was just referring to genomics, but now we have transcriptomics, we have proteomics, we have metabolomics, we have omics at all levels. So the idea is that we put all these into equations, let the computer kind of digest them, produce results, and then basically these predictions we have always to get to experiments to validate them in some way. Okay, in other words, when we get to a point in which we cannot do anything with genetics of cell biology, if there's a way of approaching it mathematically, usually or sometimes or many times, that pays off. And that's basically what happened. How do I solve this plumbing problem? Well, this was basically resolved by this lady working with the other guy, Veronica Kainaiser. And this is an interesting story that I would also like to tell you because you guys have these ideas from books and kind of TV that scientists, I mean, always have these kind of patterns. I mean, they all study a certain way and they behave a certain way. No, I mean, most of the big stories are just happening because people have personal, particular pathways. And that happens by chance. So don't be kind of formatted like if there's only one kind of scientist and if you want to do research, you have to do things in one way. That doesn't exist. She's a mathematician, and there was this meeting on mathematical biology in Brazil. She's Brazilian. She went there. She saw this beautiful, blonde, blue-eyed Dutch guy next to a poster and said, wow, he's cute. And then started making some clever questions to the guy. Eventually they fell in love. Now they're married and got kids and everything. But as a consequence, she went to the Netherlands with him. And where he was working, there's this lady here, Pauline Togbeck, which by any sort of standard, it's a crazy woman. Okay? But she's like one of the most brilliant mathematicians in the world. And she's so brilliant that her university built a beautiful building just for her kind of biological computational building. And she stopped working under the supervision of her, a brilliant mathematician, and this guy, which is one of the most brilliant geneticists developed. And the idea was like, can we develop a model that we then can test genetically? And that's what she did as a mathematician. I'm not telling you this story because of happy end. She's probably the only person I know that when she finished the PhD, not only she won a lot of prizes from this paper, but she immediately was offered a position as a PI in one of the most distinguished institutes in the world. So that tells you that sometimes when you do things differently, it pays off if it's a good idea. And tells you also that modeling and computational biology, it's a big deal, and it's going to only become bigger in the next years. So you may consider that for your career option. So what did she do? Actually, for a modeler, this is actually really basic. She did this very, very crude model in which the root is a rectangle, and every cell in the root is a smaller rectangle. These are this gray area here is the quiescent center. This is the endodermis. This is the epidermis. Actually, she kind of stripped, all the complexity which was possible to strip out of the system. She considered 4 patterns of pin localization. This one in which the pins are on the side, which are localized here. This one in which the pins are on the top, which is localized this here. This one's in which the pins are on the bottom, which is here. And basically this one here in pins are all over the place, which is the quiescent cell. Okay? So in other words, she put arrows on the model. So where you have the pins in one position, you deduce that the oxygen should flow onto that direction. She went to the literature and she used the best possible numbers for arbitrating the fluxes and asked the question, if I throw this everything into a model, will the model produce what I see in terms of the oxygen distribution? And this is the cool part, and that's when you start thinking that, maybe these things happen. This is just a normal model, just assuming that there's oxygens coming from the chute, and you start the model, and the oxygen redistributes through the model just based on the localization of the pins and the size of the cells. Okay? Nothing else. As simple as that. Okay? Conclusion. Immediately you see that there's a formation of a quiescent center here. Not because she said this is the quiescent center, but because she just imposed these conditions to the model. But as you pointed out, concentration was the issue here because she was not seeing the different concentrations as sharp as they should be from at least from the images that they are found. So she made another simulation, and in this case, she just didn't consider just the oxygen that's coming from the chute, but also that every cell produces oxygen. And we know that is the case, and there's literature to produce it. Now the new image she's got is much sharper. And there's a number of curious examples here that you could never predict would happen. But that the model showed up that these 2 cells, not the immediate one, but the next to it, should have more oxygens than this one. And these 2 should have more oxygens than these 2 in the center. guess what? They went back to the microscope and tuned up the imaging conditions, and they found that this was true. This was completely unpredictable. People were just watching for the roots for 10 years and they never noticed it, just because they were not alert to it. But when they look at the model, could it be that this is actually happening? And it does. Oh, this one here, I can't remember what it was because, I mean, the description of the experiments is made on the beginning, but it's some experiment. And finally on this one, I think that they just, at a certain point, they have the root growing and they cut the root. Like if you go there with the scissors, so in order to stop the flow of oxygen that's coming from the top. I don't know what happened here, but I did something. Or slightly something. I mean, never mind. This is just to tell you that when you have a model, you can do 10 experiments in a day. And you can change the conditions of the model, and you can generate 20 mutants in a day. And if you come to some model or some conclusion or some theory how the system works, then you just have to do one or few experiments to validate the whole thing. And that can save you 10 years of research, which was the case. So basically, they went back to the lab, and this is the cool thing. I mean, they could generate manipulations of the system either using genetics or using just incubations with different inhibitors and different molecules, and basically could do kind of a beautiful correspondence between the predictions of the model and what happens in real life. Okay? When you get to this point, then you get wild and you start doing all sorts of things on the model and making crazy predictions about the model, which they did here. Here it's not just confirmations of the model, it's really predictions. For instance, in this case here, about ablation. What if you ablate these cells? She hasn't done anything for the model. The model immediately predicts that the quiescent cell center goes up. And that's what they got from the ablation experiment, if you remember. Okay? The other conclusion is like the minimum model to explain cell-to-cell signaling and patterning in the root is that you only need to put the pins on the right position and let them flow the auxins. So auxins carry the message in the form of flux, but fix the cells into differentiation by the concentration. Okay? And this is an important conclusion because when you get to animal development, we know that it's all about morphogens. So what is the concept of morphogen? The concept of morphogen is a protein or a hormone or diffusing something that, depending on the concentration, is going to induce different cell fates on the cells. Okay, the typical example is this one here. I spoke about EDGOG the last class. EDGOG is one of the most important molecules during development. If you make a mutant on EDGOG, everything gets screwed up in an embryo, basically. And it's a morphogen. And the concept is illustrated here. So if you have a limb bud, and if these 5 dots you have here are the 5 perspective fingers from a hand, let's say if you are talking about a mouse or a vertebrate, a mammal or whatever, then you can prove that this position of the fingers, you don't know that, but these 2 fingers are made from the same progenitors, these 2 fingers also, and the thumb is made from a different area. And what defines these 3 domains of development of your fingers is the concentration of sonic hedgehog. So there's a group of cells in the limb bud that segregates Sonic the Hedgehog, which is excreted, and because it's excreted from this point, it forms a gradient. And when you have a little bit less of hedgehog, you have the middle fingers, and when you have no hedgehog, or just a fraction of the hedgehog, you have the thumb. Okay? That makes sense to everybody? This is the concept of morphogens. These cells in the limb bud, of course, are competent to read this gradient. If you put a lot of hedgehog in other cell, probably not going to see anything. So it's a combination of cells being competent to read hedgehog and then a concentration of hedgehog. Okay? Chicks, on the other hand, because these 2 fingers were fused in one and these 2 middle ones were fused in one, you just have these three patterns. Okay? How can you test this hypothesis? Very easily. You get here and say, like, you flood this area here with sonic hedgehog. And this can be done easily on the lab just by soaking a bead with hedgehog, with a protein, and just put it in contact with a limb bud. Okay? So what you're doing now here is instead of just having these cells producing hedgehog, Now you put another source of hedgehog at this position. What would be your prediction? Now you have a lot of hedgehog everywhere. What would be the prediction? A lot of hedgehogs everywhere. Well, you're looking at the slides. I should have closed this because the prediction would be that you would only have, what is it, fingers like this everywhere. Well, the prediction is a little bit more complicated. You have a repetition of the limb. In other words, a lot of hedgehog create another foci, and basically how these cells interpreted this was making duplicate number of fingers. In other words, these chickens have 2 hands in place of 1, basically, if you can imagine this. Good. Does that make sense to everybody? Now, the concept of morphogeny has been around since the 60s. But, you know, sonic hedgehog and another protein in animals were the first ones to be demonstrated to work like this. So you have a point source. The point source kind of diffuses the protein, signals to the cells around, and then differentiate to form patterns. And that's how we think a lot of neurogenesis and a lot of other processes are formed. Could it be that auxin is a plant morphogenesis? Now, of course, that implies that auxin should be able to change some genetic programs. And of course, we have to find what are the targets of auxins in terms of genetic programs. Okay? So when we think about these things, the concept that you should bear in mind is that we should look for a specific kind of protein called transcription factors. Are you guys familiar with the concept of transcription factors? No? No one? So what is a transcription factor? Anyone can tell me? Yes?
It's a protein that affects the transcription of the gene.
Exactly. Transcription factor, by definition, is a protein that binds directly to DNA to switch on genes. So when you think about, oh, I think this hormone is starting a new genetic program, you have to think there's got to be a transcription factor in the middle that it's going to be affected by that. Okay? So I'm going to jump a few genetic screens to directly one transcription factor that we know is fundamental for the formation of roots, which is plethora. What happens if you overexpress plethora, and why do we know it's fundamental for roots? If you overexpress plethora, you got roots all over the place. So this is an embryo. The poor guy is trying to do roots everywhere. No shoots. Okay, or a little bit, this is a seedling. Instead of having a nice pair of leaves on the top, it has roots everywhere. And now I ask you the question. I just used this sentence, overexpressing plethora. What does it mean, overexpressing plethora? It means that I'm inducing a lot of the protein plethora everywhere. How do I do that? Yep. You use a promoter, which
That's constitutive.
that's constitutive. What does that mean? It's turned on everywhere at high levels. Okay? So again, we get back to the promoters. So let's give an example. The promoter, the more important constitutive promoter we have in plants is a promoter called 35S. How did people come up to 35S? Because they were studying this virus, and they found out that the virus was very virulent, which means that it could produce a lot of viral proteins, and it affected all the organs. Like it could infect roots, it could infect leaves, it could infect flowers, anything. It was just a nasty virus for certain species. So people reasoned, well, if this virus can infect all the organs and produce high levels of viral proteins everywhere, then it should have a very strong constitutive promoter. That sound reasonable to everybody? So they went on and they found this 35S promoter. And that's the promoter that we've been using for the past 25 years to induce overexpression of genes in plants. In other words, if I make a construct in which I put plethora, in which I put 35S driving plethora genes, basically I have a plant in which all the cells are going to express a lot of the plethora protein everywhere. It's flooding all the cells with plethora. What do I get? A very sick plant with roots everywhere. What does that tell me? That plethora alone is enough to induce the root development. Okay? And I don't know if you saw those pictures. I mean, this is the same sort of reasoning that people did when they produced these flies that have a leg on the egg or something like that. Did you guys ever saw those pictures? Okay? Now, these plethora genes come in various flavors. I mean, all of them, when you look at the expression pattern, what am I looking here? What is this blue thing? Now you guys should start asking me this, answer me this question. Why is it blue? Why is this root blue? Do we have root blues in the, blue roots in nature? No. So why is it blue? Yeah. It's expressing Gus. And if I'm trying to look the expression pattern of a gene, what do? We're getting very quickly there. How can I say where is a gene expressed? What defines the expression of the gene? You.
RNA.
Yeah, right, but the RNA is expressed from the DNA. What makes that specific part of the DNA being expressed?
Promoter.
The promoter. So if I suspect that the gene is important, I isolate the promoter of the gene. Instead of putting that protein coding frame, I put Gus. So by making Gus promoter fusion, I can know where the gene is expressed. So here, for instance, they used the promoter of plethora 1, put it Gus, and this is the expression pattern. Here they put the promoter of plethora 2, and this is the gene expression. Look, always highly concentrated on the quiescent center, which makes sense for a protein that does roots. Okay? But now notice the thing, I mean, there's a gradient of the protein, or in this case of the expression. I mean, it does a gradient like ranging in other directions, like oxides. Does that have anything to do with, you know, like the pattern that we observe, this plethora involved on that? And I'm just going to skip this because we're short on time. This is the scheme they came up to. I mean, plethora specifically regulates the disposition of certain pins, and on the other hand, certain pins also affect the position of plethora by accumulating oxygen. Okay? Now, here's the thing now. So we're testing the hypothesis that auxins is like the thing that does the cell-cell signaling, and plethora is the thing which is triggered by auxins, and basically generates a quiescent center and generates a root. Is everybody with me? Now, how could I test this hypothesis? You know, I mean, like, if the plethora is the thing that is doing the job for auxins, or it's the thing that is receiving the signaling from the auxins, how could I test this hypothesis? What have we done on the previous experiments when we moved the pins around by mutations and genetics? Am I moving too fast for you guys? Or is this just a new way of going fast through the ideas? Okay, should I help you guys? So say like, if, look at these pictures. Say, if you have a lot of plethora in the quiescent center, you can say, oh, it's the plethora that makes the quiescent center. So one way of testing this hypothesis is like, what if I can put plethora being expressed in this group of cells? What happens? What would happen? There should be what? I mean, the idea is a bit of the other way around. Auxins induce plethora. So if I put plethora there, yeah, okay. I mean, the auxins would be accumulated there. But what would be the phenotypic result? What? A new stem cell niche. In other words, I would have a root inside a root. That makes sense to anyone? Sounds like a bit Frankenstein weirdo kind of thing. Can this be done? It can. Again, going to promoters. Man, you heard a lot of promoters. Okay? So dexamethasone. Anyone knows what is dexamethasone? Thanks God, because that's the stuff that athletes that want to cheat take on. I mean, this is a very strong steroid that makes you, like, you know, puff up your muscle if you're working hard, you know. Turns out that plants don't have dexamethasone, and the dexamethasone, I mean, I'm not going to go through all the genetics, but dexamethasone binds to a receptor on the cytosol, and the cytosol goes to the nucleus and triggers a specific promoter to basically induce the genes that basically pump up the muscles of these guys. So you have a binary system. So if you put like a dex promoter, you can put, say, like plethora, and then what you have to do, of course, is you have the dex receptor. No, you can have 35S promoter with a dex receptor. So So by expressing these two proteins on a plant, you have the receptor of dexamethasone and a promoter that will be triggered on by this protein if it binds dex. And this solves a big problem, because when you overexpress Platora, what happens? You've seen it before. What happens if you overexpress everywhere Platora? Roots everywhere, which doesn't tell you anything, right? But say, like, if you have a normal plant, like already growing with leaves and roots, everything, what this allows you to do is you go there, you spray dexamethasone, like literally spray. The dexamethasone goes inside the cell, it's a steroid, crosses the membrane, binds the receptor, goes into the nucleus, and activates whatever protein you have. We call these conditional promoters. You can trigger them on whenever you want. Okay? And in plants, you can do this with ethanol, with dexamethasone, and a few other things. You just have to spray the plants to trigger a gene. Okay? And this is cool, because then you can do these nice images. A root growing on top of an apical meristem. Okay, so the plant was developing normally. You went there, you sprayed with dexamethasone, and now what you've been able to achieve was to put a root on the top of the plant. Okay? And now we go back to our original question. Do you have a doubt? Do you have a doubt? Do you have a doubt?
Oh no, we were just.
I had a question.
Sure.
I didn't understand what was actually the dex receptor.
Platora.
No, when you're doing the gene, the receptor, and then there was something else. What was it?
So you have to put two constructs. One which is going to express the receptor of dexamethasone, and another one with the promoter which it binds, the binary system. So you have to have both components. Because if you just do the receptor of dexamethasone, then it doesn't bind anything. You have to have a promoter in which it binds. Okay? That makes sense? Okay? So you induce the receptor, comes dexamethasone, the receptor goes into the nucleus, it binds to that promoter you put there, which is a dexamethasone promoter. And then you trigger the gene. Just by spraying. Okay? Yep?
So that plant's already transgenic.
They're all transgenic. Everything that we speaking about is transgenic plants. So you have a wild type plant, you put in those two things, you got a transgenic. But you don't eat them, don't worry. And if you eat them, you don't worry too much, because as far as I'm concerned, nobody ever proved any problem with transgenic plants in feed, but we're not going to discuss that. But the companies are making a lot of money to put labels gene.
If you're expressing Platora throughout the plant using this construct, these two constructs, why does it specifically form at the
Well, it's going to form everywhere. I'm going to show you the next image. It's going to pull you out of your socks. Okay, this is a normal root. Okay? This is a root after you sprayed dexamethasone when the root was here, and then it grew this bit again. Okay? First thing you notice is that cells are much smaller, which means that elongation did not occur. But the picture that really should put you out of your socks is this one. And this is not Photoshop. Okay? This is not Photoshop. So what does this picture shows you? Or these two pictures? There's a root inside a root. Okay? Which you cannot say from looking at this, but when you stay for starch, you see that there's a new quiescent center here, there's a new columella here, and basically there's a root inside the root. So in other words, what they were able to say is that, in fact, when you overexpress Platora, that is going to trigger a genetic program that's going to put pins in a way that you have supernumerary or extra organs that you should not have in the wrong place. That's conclusion number one. So Platora is sufficient to explain why auxin works as a morphogen, and this was the first morphogen that was identified in plants, auxin. And secondly, I mean, that also explains why the cells don't elongate, because all the cells have a lot of auxin now. And because all the cells have a lot of auxin, they don't elongate, they keep dividing, and they all stay small. It's like, you know, you have an extension of the stem cell niche to the whole area here. Look at the size of the cells here, and look at the size of the cells here. They're much smaller. So it's like this whole region now is a stem cell niche. They keep dividing. Okay? So pretty much what they concluded was the following. Factors with a graded distribution can program fields of cells in the dose-response manner. And this is like, if you want, the most important consequence of cell-to-cell signaling in development. Certain cells are competent to react to certain concentrations of certain hormones or certain proteins or factors, like sonic hedgehog, or a number of other factors, or a number of other hormones that we know are important for development of cell-to-cell signaling. Okay. How is that achieved? It's achieved because there are mechanisms that put it in a specific spot of the organ, like the limb bud initial, or in this case the quiescent cell centers of the root. They concentrate the production or the accumulation of that morphogen, and that creates a field of concentration around it that creates the formation of the pattern and the different differentiation patterns of the cells. Okay? And that is exclusive of the stem cell niches. Okay? Now, we still have like 15 minutes, so let's digress a little bit on this term. But before we go that, I mean, did you guys really understood the sequence that I just got you through? There's this hormone that people knew from the times of Darwin was important to change the shape of the cells. People developed tools to investigate the concentration of this hormone, the DR5. People found, but through genetic screens, the molecules that transport these auxins. And then by genetics, people formulated this hypothesis that maybe it's the accumulation of these auxins in this group of cells that actually defines the stem cell niche. And then people validated this model through computational biology. Okay? And basically the last step was this paper. People proved that it acts at the morphogen, again through genetics. Okay? Does that apply? And the beauty when you kind of develop the theory like this is that then you get tempted, maybe this is a general mechanism, maybe everything is like that. Like every organ in a plant is generated by accumulations of auxin. It turns out it's true. So lateral root initiation, we already spoke about it. See, I mean accumulation of auxin in one single cell, and you got a lateral root, the whole thing. Okay? Go to the tip, to the apical. You saw the movie of Marovitz. I'm not going to give you the whole story because you've already watched the movie, the videos, or you should have. But you got maximum of auxin wherever you have the formation of a leaf or a flower, any organ. So apparently there's a way in which cells or the tissue is organized to accumulate auxin in certain spots, and those spots are going to develop new organs. And this is the variation on the theme. And this is very interesting because if you go under the dirt, the accumulation of auxin is generated by what we call a reverse fountain. So auxins go by the middle, and they come back by the periphery. But if you look at the shoot, it's the other way around. Auxins come by the epidermis, and basically come back by the center. So it's a variation, but the concept is the same. You accumulate auxins in a couple of cells by cell-to-cell signaling. These cells, they change, and they become a stem cell niche and form a new organ. Okay? I could tell you the same story with limbs or any organ or many organs in animals. Not in canalization. Well, this gentleman just basically was one of the few that solved the problem that you also observed in the other films from Marovitz, which is the spiral phyllotaxis. I'm surprised that Marovitz did the whole video and never cited the Da Vinci Code. Who from here saw or read the Da Vinci Code? 3 persons? Oh, more. Okay. So, what is the Fibonacci series, or what is the Fibonacci number? What did you learn from that on the Da Vinci Code? You didn't learn anything. Yeah.
Is that when you like add the previous numbers together to get the next?
You get kind of you make what it's called a mathematical succession, and that by magic always produces the same number. Okay? And this is the cool thing, as you look in nature, this number is reflected in a number of things, and that's what in Da Vinci Code, because it's a lot of speculation about nothing, because this is, just trivia. So when you look, say like at a plant, I wanted to buy a plant this morning and I forgot. And if you look at the consecutive leaves, or flowers, and if you measure the angles between every adjacent flower, you kind of think, man, this guy's no geometry, because the angle always tends to be a Fibonacci number, 137 degrees, okay, which is one of the perfect solutions of a Fibonacci series. So you kind of think, oh, plants know mathematics. How can they calculate this so easy? Turns out that it's just evolution at work. Turns out that mathematicians on the 30s, in 1930s, proved that if plants put leaves at this angle, this is the perfect solution for all the leaves to get the optimal light. So this gives them an advantage. That's how they learn mathematics. Bees also know mathematics. When you see the beehives, it's always hexagonal. Why? Because the hexagon is the form, the shape, that gives you more volume with less surface. So they're sparing on walks to put in the honey. That's why they learn mathematics, because that gives them an advantage. Okay? So the thing here is, the sphelotaxis is also generated by this oxon accumulation. And what this gentleman did was, again, experiments that were done in the beginning of the 20th century. So this is a pin 1 mutant. No leaves, no flowers. What has he done? An experiment that was firstly done, I think, in 1925. Pick up a bit of wax, soak the wax in oxin, and stick it to the stem. What do you have? Just the application of the wax with the oxin, or just the application of the wax. What happened to my mouse? Oh, there it is. Just the application of the wax with the oxin is enough to induce a new flower. Okay? And then he got, this experiment must have been fun. I mean, I imagine this guy, the whole day looking at the stereo microscope and just putting wax around every possible part of the plant. In other words, he could do, leaves which are basically round around the meristem. I mean, he could do leaves with 2 parts. I mean, he could do all sorts of weird kind of monsters just by playing around with auxins in the pin 1 mutant, because the pin 1 mutant does not bring auxins to the ship. Anyway, he was not just doing that, he was looking at the RNA of certain genes, he was looking at the pins and everything, and he came up to this model. So basically the model that he came up to is auxins are brought to the top by the epidermis because the pin 1 is located on the top of the cells in the epidermis. But guess what? It gets to a point in this part of the meristem in which there's no pins. So auxin gets accumulated there. And when it reaches a certain threshold, it first starts doing pins to kind of diverge the auxin flux and starts a new organ. Okay, and this is the beauty again of mathematics. And again, this was 3 papers that came out on the same issue of PNAS. Again, a very relevant journal. When you see papers that came out in this journal, usually they means that they are very good quality. I mean, you also have problems sometimes, but that's the general line. And 3 groups simultaneously, including the one from Elliott Marovitz, produced mathematical models with the following. Again, they just tried to do a physical model of the apical meristem, put the pins, kind of arbitrated values for the fluxes of the auxins, and see if that creates a pattern. Now you saw the movies that Elliott showed in that video that you guys were given. And that's basically coming out of this paper, and that model is lousy because the best model, of course, it was not is, so we wouldn't show it. I mean, show this model, although it's not so beautiful. It's this one. And now what you see is the following. There's no constraints in the model. See, where you have more, the green shades reflect the quantity of auxins. The red traces reflect the position of the pins. You start with just a group of cells, and you impose a certain division, localization of the pins, accumulation of auxins. And if you measure the angles of the new leaves which are being formed, now you have to imagine that these things are growing to you and you're like stepping back. Okay, that's why they disappear on the back. It's just because the model, was done so that you don't see the whole plant, otherwise it would get confusing. But what you should think is that it's constantly growing, producing one leaf, producing another leaf, producing another leaf, producing another leaf, etc., etc. Just by throwing into the model these basic concepts: concentration of auxin, localization of pins, cell division, and the shape of the organ. Just by magic, the Fibonacci angle shows up. Okay? So it's a constraint of the system which is encoded basically in the size of the organ, and these very fundamental mechanisms that all the plant cells are capable of evoking at some point. And this also explains why when a cow comes and eats a plant, you can have another organ, because at any point it can reproduce and reiterate this protein. Okay? Does this make sense to everybody? Are you guys tired with auxins, or just giving you too much information? Be honest. I can stop here. No, I can't. Okay, just to finish, just a few kind of global concepts, and I'm nearly done, I promise. First, you have stem cell niches where you need to develop new organs. Well, we studied the case of the root, so these are made around these quiescent cells, center that kind of out the cells around it for differentiation, but make them divide, and this division that will create the different patterns of cells in every direction. You asked the question, what about the shoot meristem? The system is not exactly equal, but it's almost equal. You have a group of cells which are basically organized in this case of organizing center, and this group of cells is basically priming these 3 cell layers on top of it, not to differentiate, but just to divide. And when they divide to the sides, and then these cells differentiate into the different cell types and the different organs. And in the apical meristem, we know very well the cascade of things that are needed for this. I'm not going to go through all these names and all these things, but for instance, this clavata tree, it's a small peptide, so it works like auxin. It goes from one cell to the other and establishes a gradient of concentration. The cytokinin is another type of hormone in plants that also does that effect. Okay, just to summarize everything, because I just give you an example because I thought it was a good example and we can go from the observation of effect and tell the whole story. Let's just put in some concepts, because you love concepts and you love to, and then they're important. So everything that I told you about until now refers to a type of cell-to-cell signaling which is called paracrine, which basically stands for soluble molecules that move between cells closely located to carry information. That's what auxins do. They do from cell to cell, and you know the concentration of molecules that actually moves defines the response. But there are other types of cell-to-cell signaling, and the one that I should mention here is what we call justacrine. Now the justacrine, it's not a diffusible factor. It's actually physical interaction between things. Okay? So there's a receptor and a ligand. Say, like for instance, in this cell, you have a receptor called Notch, which is like the most important receptor in development. It's responsible for all the differentiation of the neurons and all the arteries and all the veins. It's absolutely essential. If you knock out all the notches, you don't have a nervous system, you don't have veins, you have like, you know, a mass of cells. And the reason why neurons and angiogenesis, the formation of new vessels, occurs is because some cells kind of signal to their neighbors, oh, you got to be a neuron. And they do that by this system. One of them has a delta, the other one has a notch, and when they get together, they kind of trigger a cascade of events, and the one that has a notch becomes a different cell. Okay? So if you have an undifferentiated tissue, and this is triggered, then the one that has a notch becomes a neuron. And the other one just becomes mesenchyma or whatever. Okay? That makes sense to everybody? But there has to be a physical interaction between these two proteins. Also, in the justacrine signaling, I mean, your Norm asked me to make a point about this because I think she spoke about gap junctions. Did you guys spoke about gap junctions? Okay. So gap junctions, again, it's a specific example of justacrine signaling, but a little bit more dramatic in the sense that the cells actually, the cytosol becomes united by these communicating channels. So that you guys know, I mean, plants also have these, actually in a much larger extent. So we call these channels plasmodesmata. And as you can see here, I mean, 2 cells can be connected by dozens of these channels, which literally means that a whole tissue can be communicating through these channels. So diffusible molecules or signaling molecules can go from cell to cell through these channels. The internal structure of these plasmodesmata is very complex. But just to say that you can go, I mean, if they're closed, if this structure is closed, I mean, only very small things can go, amino acids, sugar, stuff like that. But if they're open, big proteins can go, RNA can go. And what is more important, viruses learn how to use it, And virus could go. Virus can get here, they hook to this complex, they open, and they go through the plasmodesmata. And they can infect the whole plant just going back to plasmodesmata. Viruses are damn creatures. They really learn how to do tricks very fast. Okay? And I guess this is it. So I hope you enjoyed it. Anyway, you have my contact. Some of you already showed that. If you have any doubts about the material, I put in 2 more documents regarding more specific animal-to-animal, cell-to-cell signaling mechanisms. If you have any doubt about them, don't hesitate to